it will be very difficult to communicate all of them, but I will try my best to communicate some of the areas where the institute, at least the department, has contributed in terms of research and activities. Well, as you know that many of you may agree with me that being a surgeon, you see, the researchers are quite less, relatively less. If you see the grant received, you see, not only in our country, internationally, the surgeons, they get a less research. I mean, if you see that the surgical people, they have a less research grant, rather less interested, rather, I will say, in these areas, considering the research grant they get, you see. It is because of probably their more involvement in clinical sites, but there are very many innovative research work. In spite of that, they have carried out, you see, including basic researches. Now, let me share my screen, you see, then it will be nice for me to communicate you see, yeah, probably.
Yes, sir, we can see it. Please.
There are a lot of things we could communicate to my young friends, but it will be, because of due to time constraint, it will be very difficult. So once again,
sir, can you put it on full screen?
I'll do it. So once again, I say thank you for giving me an opportunity to interact with you. So these are some of the research areas which I'm going to discuss in another maybe some 45 minutes or 50 minutes of time. See, what basically research is a careful and detailed study of a particular problem. So to my friends, young friends, so research means there should be a problem. That is probably more important. There should be something concerned which bothers us. And now then, what is the existing knowledge available with that problem? What we can do to solve the problem? That is probably what it lies in. It's a basic instinct of our human nature, even in many animals also, that we are curious. That makes us to find out something new or something validate where we stand. It's a pleasure of finding things. That's what we call as a research. So as a surgeon, as I told you, that most of us, we are involved with doing some sort of a patient care, operating patients, treating patients. So the chance of curiosity in dealing a particular problems are relatively less. So it consists of 3 steps. As I told you that you pose a question, there should be a problem. Then to solve the problem, we collect some information, something we collect to how to solve that problem. And finally, we try to analyze this data and the information, whatever we receive, and then we try to find out an answer of that question. That what is the basic applied researches. So these are a very now basic research so therefore any scientific things, whenever we go in a science, you see, we carried out some basic researches which enable us to know how the things are going on, how the nature take care of it. To be honestly speaking, basic research are quite difficult, very difficult to answer these things. It seems to be very simple, but quite difficult to answer. The transitional research, once we understand how it happens, how they can be translated to solve a problem. Clinical research is something where we discover and innovate for this their application in patients or in making somebody to remain healthy in a particular group or in a particular condition. That it comes in a clinical research. So these are some of the basic things what I'm trying to tell you. Basic research results in new knowledge. It is responsible for many research breakthroughs in different fields of study, and it often considered as a important pillar in technological and innovative solutions. The findings of fundamental research are extremely useful in expanding the pool of knowledge in different disciplines. Yeah. Say, in a in a, in a circumstances of a genetic research, it not only helps in to understand, in many areas, you can identify a person's by a gene, you can find out, you can develop certain drugs, medicine by using a particular gene. So look at, it's a basic to understand the structure of a gene, their arrangement. So you can do so many things, many things you can do. So therefore, basic research is very important, to understand for applied researches. That the last is very important, it takes several thousands, hundreds of years, and also that is very important. There are very few genius people. They contribute in understanding this, how the nature works, means in true basic researches. Therefore, it takes several years, hundreds of years at times, and there are very genius people, they really involved in basic research. That is probably more important, aspect. Just take a very simple look on it, It took hundreds of years to understand, to explore the genetic structure, gene, their DNA sequences, how the protein synthesis goes. So in developing an insulin, what we use today, as we all know, that there is a particular segment of gene is responsible for a particular protein, that is insulin, and that insulin, a segment of gene is responsible. So that is been separated, that is done. They are then put into in a E. coli usually, the bacteria, and now the bacteria now start producing a pure insulin. That insulin is now being used. Simple. Hundreds of years and many researchers to find out this particular sequence to develop a thing. That's probably a basic research, what it takes a time and what it ultimately, This is one such example. I was area where I was, my co-researcher was from different field. He was working with Manju. He was using he did the same technology, He identified the gene which is responsible for producing this color of this Manjushta, and then he put it in a E. coli to generate, a pure dye, a color which is used for making color for printing, for staining, clothes, which is many a times we use as an Ayurveda. So this is a natural dye is being produced by the same technology being used. Just I'm giving an example, why that how diversified, this a basic research can help, At one side, it can give you an individual person, at an individual and insulin, a drug, same way it can give you a color, produce it. So many things can be done by changing, this genetic, understanding the genes. If we talk about our own science, now in Ayurveda, as we call it, basic sciences. So it is really very difficult to understand the basic understanding on which the Ayurveda stands, you see, the fundamental philosophy of Ayurveda. To my mind, we truly do not know anything about the fundamentals of Ayurveda on scientific basis, or very little is known, you see. They are fundamental. So just say the human brain, and if we see the mana, see probably the mind, man, it's really a very basis of our Ayurvedic philosophy and many diseases and our own, you see, how the human being developed, you see. So this scientists, you see, now consider that human brain intelligent enough to fully understand itself. The answer is no. The researcher says the mind cannot possibly study itself. हमारे दिमाग में इतने बुद्धि नहीं है कि हम अपने दिमाग को पढ़ सकें. That probably what I say, you see. So therefore, it is very difficult to understand, to scientifically evaluate, to study our own mind by the intelligence, whatever we have, you see. So if we could not understand our mind, how could we could study our own mind? How could you study, you see, Atma, Paramatma, the things which are basis of Ayurveda? Therefore, the fundamental, the research in Ayurveda is really very difficult. And hardly we have done anything on fundamental research of Ayurveda. And can science truly explain these things, you see, like consciousness, love, maturity? Probably it takes a lot of more time to understand it, you see. As we all know that, you see, this Ayurveda is based on this Man, Panchamahabhut, Dhatu, Srot, Vikriti, Sar, Ojas, Rash, Virya, Atma, Dosh, Mal, Prakriti, Satya, the Agni, Gun, Vifak. Hardly we have any that, you see, whatever we put together right now, you see, are nothing but the collections which are available in different texts. And then, or the different texts they are available, we review these things. They are secondary outcome, you see, not to explain, you see, on scientific basis that what, you see, this Panchamahabhut, Srotas. So like this, you see, so it become really difficult to do a basic research on this area. Recently, there are few research areas what we could see on the Prakriti. And this is genomic study of Prakriti. There are few, they are very, the tip of the iceberg, what you could see. Initially, there are few studies has now started, and now we could see some of the fundamental research on these areas. Well, you see, now I'm coming to Shalyatantra especially in particular. Therefore, there is enough, you see, scope to understand the basic researches on these areas, which will explain, see, the understanding of applications of in many, all disciplines of Ayurveda, that may be Kaya Chikitsa, that may be Shalya Chikitsa, maybe many all other disciplines of Ayurveda. Now, if you see that in Susruta's contribution in understanding of disease, we call development of a disease is Satakriya Kal, you see. Well, that's a simple understanding that there is a disease, starts, chai, dosh. So we do not know in terms of the scientific validation of doshas. So what actually accumulate? What actually prakop? What actually vitiates? Is there something? Is there any biological entity? Is there anything, some chemical entity? So these need to be answered, you see, in course of a time, to explain these Satkriyakal in due course of time, you see. So it is one of the most, you see, important contribution given by Susrut, that there are 6 stages of development of a disease. These Satkriyakals need to be explained in terms of some biological, physiological, or chemical entities, if it is possible, you see. They need to be understood and a basic scientific understanding and researches. Well, we could see that most of the Shalyatantra is mainly it is by Susrut, contribution of Maharshi Susrut, and this is one such a, and he is also known as father of surgery, and we also, most of us internationally, he is known, we call it a father of surgery, but internationally people take him more commonly call it a father of plastic surgery. He has given certain basic principles of plastic and reconstructive surgery. These principles are being opted to, these principles are being taken to conduct many new technique of plastic and reconstructive surgeries, which we have realized much later, you see. These principles are on the philosophy that it's a plastic surgery, as we all know, that it's a establishment of establishing a relation between flesh and blood. So where we try create a new relation between a a piece of a tissue with the blood, so that it can survive, you see, where it has placed. And he has given a principles, mentioned a principle that if the blood is not pure, if there is a inadequate blood, if there is a dushta rakt, or if there is an excess of blood, in all these circumstances, there should be a rejection or there will be failure of grafting. That was his principle. The other principle, whatever he has made, that you see, if you take a part of it, you can a flesh can survive, you see, if you keep a pedicle where there is a circulation and the reconstructed part get the blood from through the pedicle, so that it should remain alive, you see. If you take you can remove a part of a tissue from cheek, so it will remain alive if there is a pedicle is there, you see, which can be used subsequently for construction of ear lobule or nose also. That was his concept, you see. This concept was being taken for reconstruction of nose by him at times. Why I talking these issues, you see, the Institute of Medical Sciences did a tremendous research work, you see, to validate that how the microcirculation develops once, say, this pedicle, it maintains the vascularity, as well as once the graft is shifted and a microcirculation is established in between the grafted area. That was a basic research which was carried out with the help of plastic surgeons and Ayurvedic people in the Institute of Medical Sciences. When we started, say, the institute was started, it was in around 1964. So it was in sometime in 1970s. The research, the Banaras University was one of the first university or the institute where the research laboratory was established in the name of Surgical Research Laboratory. Most of you will be pleased to know that this research laboratory was initiated by Professor K. Udupi, the then director, and was occupied mostly by the researcher from the Ayurveda, and it was also supported by a very eminent researcher and surgeons from modern system of medicine, and they worked together. This work was established, the microcirculation, microangiography was carried out to establish how the vascularity was established on those in between these a pedicle graft. There are two sources, one from the nasal side as well as these how these micro vessels they take place and they survive these things. You see. So these principles of Sushrutas were re-established at our center. It was quite long back, you see. So this application could be taken. It was Sushrut who took this flap from the nasal side, from the forehead, from the cheek, you see, to reconstruct the nose. This was relatively a clean area, but these can be taken other areas also to cover an infected area which is covered by. We took the same principle to take a pedicle flap from the thigh. Normally, these pedicle flaps are taken away. The area is covered by a split thickness graft, a thin graft, you see, which are sometimes cause more difficulties. But if you see that the scrotal sacs which are damaged, Ayurveda also says that if there is a scrotal sacs are damaged, they should be placed properly in the thigh or abdomen. And the other way, we can do it also. We can take a pedicle flap, keeping the pedicle, the blood supply intact, and reconstruct the scrotal sac using the same technique opted by the principle. Now, question arises, and look at you see the reconstruction could be done very successfully using the technique given by him. Now, this technique need to be verified, validated with other contemporary techniques. That is a kind of a research we need to carry out, you see, in clinical research, where this kind of a technique which is opted by him, by Sushrut, is it applicable or comparable to other kind of a, so therefore it needs a comparative clinical trial on these areas to validate the implications of this kind of a technique used in. So therefore many such conditions can be done, you see, by opting the Sushruta's technique in plastic and reconstructive surgery. I would take one more example in these areas. What you could see, it's a incontinence of anal canal where due to damage, you see, of surgical damage, the anal orifice becomes too narrow. Conventionally, even today, what we do a repeated dilatation, or what we do, we take a flap from here and put it inside to make it wide. We have taken a different stage of practice, you see, opting the Sushruta's principles. What we have made, we have taken a pedicle flap, again, what Sushruta has described, that if you take a flap of tissue from the area and with a pedicle, it will remain alive. The same pedicle flap is now, as you see, separated, and the stricture area dissected and placed into the anal canal to make it wide, you see. And at the same time, the new skin, the new tissue will remain alive, and there will be no more stricture, you see, will develop further. So therefore, these principles, which is opted by Sushrut, can be useful in, this can be replicated as a type of a principles, you see, where what was described in Sushrut in correction of many diseases. And I feel that though there is no comparative study of conventional treatment, whatever we opt for treatment of anal stricture, and there is a Sushruta's principle and practice in managing anal stricture, though he has described these principles in the reconstruction of nose, but the same principles and technique can be opted in reconstruction of stricture of anal canal. So these are some of the, these are some of the things. So now let us consider these two issues. One is a basic understanding of a basic science researching, and the other is a applied, you see. I'll take you a small video. It shows you that the swimming, you see. This swimming, if someone is swimming, and see this, there is a displacement of water, and there is a lot of hydrokinetics in swimming, you see. So every movement, there is a lot of the fluid is coming, the force, there are several things are there. Understanding this swimming through hydrodynamics is a basic research. But how to cross, you see, from one place to another is a application, practical application of these researches. And probably those who are swimming, they do not understand these basic researches. So the same thing happened with us, you see. See, in our system of medicine, we have been there are a lot of basic fundamental principles are there. As a surgeon or as an Ayurvedist, you realize these things, but we have never been permitted to do swimming, you see, only to understand because surgery is being prohibited in surgical practices are being prohibited in Indian system of medicine for several hundreds of years, you see, and only we remain limited ourselves of understanding the hydrodynamics, you see, how it happens. But how to do it probably, until and unless we are being tuned in both these things, we may not able to win the battle, you see. Therefore, a basic researches as well as clinical practice, both are equally important in understanding as well as promoting our system of medicine. Now, clinical research, as we now, if you see that more documented clinical research was started, it is sometime quite long back, you see, it was 600 BC rather, I would say. So when it was in Egypt, when Babylonian civilization, when the king, you see, it was recorded in Daniel book of Daniel, where the king has given meat and wine to one group of people and then bean and water to another group of people, young people, to see that who is more powerful, will be more useful in fighting, you see. Therefore, and what he found that it was the bean and water, that is vegetarian people are more powerful than those who eat meat and wine, you see. That was the first recorded observation found as a clinical trial. Nowadays, there are a lot of clinical trials are going on, and this, as we all know, that the clinical trial has a phases: phase 1, phase 2, phase 3, and phase 4 clinical researches. The phase 1, before that, there is a one more what we call a phase 0. Sometimes we call a laboratory or experimental work in animal research work, we call it. The safety, efficacy, safety initially in phase 1, as we all know that a very limited number of people the drug is given. In phase 2, there are more people are given to understand the safety and dosing also. In phase 3, we also try to understand, you see, efficacy, adverse effect, and they are finally, once it is found that they're efficacious in a particular condition, then this drug is given for an approval. You see that these are the normally a clinical trial goes for these these 4 phases. You see, I'd like to say some few things, you see, regarding that. Always it is believed that that there are a lot of very mature people are required for doing a good research. It is not always true, you see. I feel it is your curiosity which makes you to solve a problem or to discover something new, you see. We all know Virchow's gland, you see. Virchow's gland, which is a palpable left supraclavicular gland, which is usually present, you see, in cases of a stomach carcinoma or many times other cancer of abdominal organs, which ultimately spread to the lymphatic glands, left supraclavicular lymphatic glands through the lymphatics. This we know quite simply today, but you will be, I'm sure many of you know it also, that it is the Rudolf Virchow who discovered it in sometime 1940s, probably 1944. He is also considered a father of pathology, you see. He was a student of anatomy at that time, a resident, and what he observed, you see, not a old man, he's hardly 20 to 24, 24 to 25 years young resident, and he observed that in dead bodies, you see, in cadaver dissection, what he found that in many cases of stomach cancer, the lymphatic glands get, the lymph node get enlarged. Then he explained, you see, how this happens. And that's why this gland is known as virtuous gland, one of the important clinical signs, and to understand the metastasis of stomach cancer to the supraclavicular area. So look at, you see, this is not somebody, it's your ingenious, it's your curiosity which makes a research, you see, or a discover something new, you see. I will take you another example, you see, because my teacher, he used to speak a lot about this, you see. It is Fogarty's catheter. You see. When he was a young, sometime, you see, as you could see over here, that it was in 1934, he born in 1934 in in America, and initially he was working as a some 14-15 years boy in a hospital for cleaning surgical instrument, and he saw many people to operate, surgeons to operate, and he saw that there are a lot of surgeons, they used to operate, you see, this deep vein thrombosis, opening the limb, and then they remove the clot by forceps, and then it causes a lot of, you see, damage to the time, anesthesia, pain, and ultimately many people die also. So in his mind, you see, he thought, it was his young mind, and he found that can do something better. Yes, then he decided that he will go for medical education, and he did accordingly. In 1960s sometime, you see, before that, he found that he could make a small nick into the vein and pass a catheter into the site where the clot is formed, and then he developed a balloon like this, and then he pulled it like, and the clot comes out, you see. Initially, he did it with the help of a small, the gloves, you see, what we use, the fingertip of a gloves. He tied it in between this, and then acts as a balloon, and that balloon helped to remove the clot. I'll show you a small video. Now, by this, you see, he become then, his work being now criticized, you see, and everybody criticized, देखो ये पता नहीं क्या लेके पहुंच गया ये blood clot निकालने के लिए. So it was the people used to criticize, the surgeon used to criticize when he started doing this job, but finally people realized that, and in 1956 at a university to carry a medical degree, and Fogarty was completing his residency surgeon, and the head of the cardiologist division, he convinced that this catheter has value, and then the Fogarty's patented his device. Now he's a billionaire, and he established an institution for developing new technology, new devices. More than 160 patents are in favor of him for developing instrument. So look at, you see, that it is not always that a great thing is required for developing something new. So all are quite young brain, young people, they develop a lot of things in understanding it, you see. So that's a Fogarty's catheter. I kept this because to my young friends, look at, you see, that's a clot, you see, and that's a catheter, what he passed, you see, and now see this balloon is inflated, and this clot is now pulled down now through the small incision. That's a vein, you see. So what you could see that, see, normally otherwise you have to lay it open the entire area, you have to cut it, and then you have to remove these clots by the forceps, with the help of a forceps. But it's a simple device, you see. So this research has made many things simple, you see, to add, you see, minimize, you see, save lives, you see, save difficulties, and which improve the quality of surgical procedures and life also. So that's what I said is, it is not always necessary that you need. Now look at, you see, that we need to consider that what are the areas where we researches in Ayurveda, especially in Shalyatantra, can be conducted. The Sushruta Samhita, one of the most popular textbook on Shalyatantra. Do you know that when this, as we all know that from our text, is the Kashiraj Devadas Dhanvantari and his disciple, he you see, communicated the Shalyatantra to Sushrut in form of a question and answer. And Sushrut, you see, now that he wrote subsequently as a text, and that we know as a Sushrut Samhita. We do not, we actually, there are a lot of controversies about Sushruta's period, time, who was he, what is his time. There are a lot of things, you see. We do not able to come out. We could we do some kind of a work, research work on these, you see. It is not only a theoretical research rather than to understand who is Sushrut, the family background, where he from, the period of Sushruta and Sushrut Samhita. Is Sushrut Samhita written by Sushrut or several scholars have contributed periodically on a small book which was initially written by Sushrut? So there are many questions they remain, you see. How to understand these things, you see. Maybe a text which we find out, the text from that we can see the instrument whatever he designed, which was made up of a metal, still do this 600 or 500 BC, India has a technology in those days to develop the metallurgy in such an extent. Is there any excavation material which are available? We do not know that there are any such excavated material which are available, which are made up of steel. As Susu described, that the quality of steel is a razor sharp. He described that the surgical instruments can save the hairs also. They are so sharp because of. So could India has a technology on those, this metallurgical technology on those days, that there are surgical instrument by 2600 years before? So these are the questions need to be find it out by answering these period of a Sushrut. If you say that, so only merely saying that Sushrut belongs to a period 2000, 1000 BC may not be sufficient enough to convince all the people all the time. So we need certain substantial things which we need to collect through researches. Therefore, a literary research, a metallurgical research, excavation of material, they're all together is necessary to you see, to understand the period of a Sushrut, what we could see, you see, here also. So therefore, there are evidences which are required to gather, you see, from different sources to say it, you see. We did a lot of work on surgical instruments. As we all know, Dr. Vijaya, along with us, along with the Department of Metallurgy, we is the surgical instrument described by Sushruta are even useful today for performing them are quite useful, or some of them are quite important instrument by instrument. So these are a great task to be done by us, you see, so that we can validate. There are many surgical instruments today are being taken away from our own resources, are being now renamed by other people by their own name.
Sir, kindly unmute yourself.
Okay, now am I audible?
Is
There
it?
is some problem probably with the internet.
जी सर सर दोबारा शेयर करेंगे आप पावर पॉइंट को
Yeah, it is. Okay, I have to share it again. Sure, I am doing it. I don't know. Yes. Is it now visible?
जी जी सर
Okay.
सर कैन यू मेक इट इन फुल स्क्रीन
Yes. Yeah. So there are many such surgical instruments. They need to be validated, you see, and their applications to be made with the help of other departments, especially with the. So we could see that there are many surgical instruments. They can be redesigned with a bit of a modification and can be used even in today's surgical practice. This is one such instrument is being used even today. Now other surgeons handle surgery also. So this is one such area what I am trying to do it. You see that yes, we could do some good research work in these areas. These instruments, their applications, their validations also. Well, I'm sure you see this type of clinical trial, randomization, blind, open label. I'm not going to discuss all these issues because they will be taught by basic researchers, but these are all applicable to surgical practices also. This randomization, why it is required, blind, open label trial. I feel these are a discussion to be made sometime, you see. And I'm going to talk some of the basic things of some of the researches on certain areas which will help us to understand, you see, the areas where we could contribute in areas. One of the areas is renal urinary stone diseases. It has a philosophy in Ayurveda, how the stone develops. Also in modern system of contemporary system of medicine, that we found that the stones are formed not, it is formed in the kidneys. They are in the somewhere in the renal tubules they are formed, and there is a microlith in the kidney tissues. There is a damage in the tubules, and these tubules there is a crust formation, and then on which there is a crystals are deposited, and these crystallization they ultimately form microliths, and these microliths are gradually get bigger and bigger, and formally it forms a, in nutshell what I am talking to it, you see, there ultimately a bigger stone is formed. So similarly, there is a philosophy in Ayurveda also. This along with this, they also say that the urine, the stone, the dosa comes along with the urine. Not they are where deposited. You see, they are. That's not. You see, there are two philosophy. One is just stagnation, and if you put something stagnated, even a clean water in a pot, there will be sedimentation and there will be stone formation. Then there will be, if there is a stasis, you see, the stasis called stone formation, then abnormal crystalloid and colloid combination. These are the two philosophy of stone formation given in Ayurveda. So along with the urine, the pathology factors also enter into the urinary bladder. Means the disease starts somewhere in the renal, not in the kidney, not in the urinary bladder. It starts. We know there are a lot of plant-based products are being used for treatment of urinary stone. One such is a gakchur, which is extensively studied as a in the treatment of urolithiasis. A stone can be produced in animal, what we call a experimental study, which are not included in clinical study, where a part of a clinical study where the glass beads are say embedded in the urinary bladder of an animal. You could see that this blood, the stones are formed so beautifully under and there is a glass bead inside it. It's a foreign body on which the glass beads are formed. See, once if you give this plant Bokchur in a treated plant, in a treated group, see there is a group, that's why I tell you that there is a control group and there is a drug treated group, which is a standard treatment, which is a research methodology. So what you see that the stones are become very fragile. They are smaller in size. And on the contrary, those were in a control group where no drug is given, so the stones are bigger in size. They are much bigger in compared to a control. In a trial group, you can see the stones are almost less in number. Therefore, it suggests, you see, that it reduces the crystallization. I mean, the drugs helps in reducing the deposition of crystals into the stones, you see, in the beads, in the needles. So therefore, it prevents crystallizations. The same, you see, that can be used in prevention of stone formation, in making a urine which reduces the chances of stone formation. The same pathology and the same principles we have taken to treat some other conditions where normally if we in treatment of obstructive uropathies, a stent is being used today and nowadays to what we call double J stent or DJ stent, which is placed from the kidney into the urinary bladder. And so that the tract remains patent if there is a obstructive uropathy. But the difficulties in this, if you keep it for a period of time, it causes sometimes it displaces, and one of the major problem, it blockages by incrustation. The salts get deposited and it removes. So it get blocked. So the purpose is lost. So now, if we use this drug, you see, for treatment of this kind of a pathology, so far there is no drugs available today to reduce the infestation of this stent, you see. But you can use this drug for removal of infestation, and you can keep that stent quite a long time, more longer time than the control, you see. So this drug is useful in treatment of formation of stone and in failure of crystallization, and therefore it keeps the stent patent for a longer duration of time. That was a study which we carried out. You could keep. So the application could be much more different than simply removal of stone, what we say today. So the other area what I am going to talk, that wound and its management is one of the important aspects of surgical practices. And there are a lot of things could be done in wound management in Ayurveda. See, the basic objective of a wound management to convert a bad wound to a good wound in a simple meaning, you see. So in terms of a terminology, what you could to convert a dust brand, a bad wound, dust means bad, to a shuddh means a good wound, you see. So that's the basic, and for which there are different procedures are there. So a research work could be taken on the procedures as well as the techniques as well as the drugs which are being used in these procedures. You see. In a nutshell, we could see that converting a bad wound to a good wound, we call Shodhan Karma, Brana Shodhan. And once the wound is become good, how to make it heal quickly, we call it Brana Ropan Karma. So I say take this opportunity to convey you. When I joined my department, my all teachers, they are really extraordinary people in wound healing because they did a very basic primary work in understanding of wound healing. As most of you know that vitamin C, the role of fibroblast in maturation of vitamin C, these fundamental works are carried out in our department by Professor P.J. Deshpande, Professor Udupi, and then other people are also. They contributed immensely in fundamental understanding of wound healing. These are the basic researchers. They also did an excellent study to demonstrate which are the drugs to whom we call Shodhan Dravyas, how to score them as a Shodhan drugs, how to identify which are Ropan drugs on the basis of scientific, on the basis of their proliferative works on the basis of their wound cleaning activities. So they try to categorize, you see, drugs to whom you call Sodhan Dravyas, to whom you call Ropan Dravyas. Anyhow, so this the basic objective of management to convert a bad to a good wound, you see. Once it is a good wound, then you need some drugs which could cover this area quickly, we call Branasodhan. We did some work using a plant-based product, you see. A Panchavalkal is one such, you see, which you have taken as a medicine, as a drug for treatment of. It shows, you see, inhibit microbial growth, it proliferates new blood vessels, which are some of the important. What we did, you see, in these, we did a say microparticles, nanoparticles of these drugs are being impregnated on a biodegradable template. What I'm trying to say these things, this is a drug which is impregnated, and gradually this impregnated drug slowly, these are the microscopic size of these drugs, this slowly it release and it disintegrate by itself, you see. So this is the beauty of this, and it helps in proliferation of cell growth. These are all experimental studies, and the drug is taken by the cells. You could also see that the Panchavalkal is being taken up by the cells, and these cells, see, it's not simply that it demonstrates these drugs are being taken up by the cells in cell culture. They proliferate much better, you see, in compared to a standard group. They release these drugs quite a long time, more than 72 hours. Experimentally, they show a better healing. This is how it is being prepared. And once you use this patch, you see, it helps in wound healing. And you need not remove the dressing material if it is not exudated for more than 72 hours, because it's continuously poured the drug. My question is, the submission is that this is a basic work on which it is a Panchavalkal, one drug which cleans the wound and helps. We can add many more kind of drugs, or we can take a study on this model to understand, to create many more such kind of drugs. We can develop a patch which can remove the dead tissue much quickly by using the same technique. We can use some drugs which can Branaropan, which can use for faster healing of things. So these basic works will help us to create many different type of wound dressing for different kind of a wound, you see. So that's what I try to say, that you can do many more research work on these areas. There are more than 100 plant-based plants are there which are being described for treatment of wound management. But the important aspect of these things, you could see that there are a lot of different people, they work together, you see. People from material science, from biotechnology, from pathology, and of course our clinicians, surgeons, it's not a one-day job, you see. It took a long time to carry out this entire work, you see, and with the help of different people. So this clinical trial, carrying out a research work, a outcome. See, with an outcome, it takes a lot of time, lot of people, and of course, funding. The money also is equally important, you see. Without money, it will be very difficult to carry out any research work. So therefore, you need a funding for taking out these kind of jobs. I will complete my talk. You see, there are many more things we could discuss, but I will just talk. There are technologies also, they help in you see, which can improve the conditions in the current practices. You see. We know mostly there are a lot of good work, research work has been carried out in Char Sutra therapy in our department. The chemical analysis of char, what is basically contained, then how to prepare, standardization of char preparation, Char Sutra preparation, and various other preparations. We all know that, you see, these complex fistulas are very difficult to handle, and conventionally we put a Char Sutra from one end to the other end, which cuts and take time to complete it. Basis the remain same, but we have redesigned, you see, this process with a continuous research program, So what we have made, you see, we could treat a simple complex fistulas more effectively. You could see a small video for this, you see. What you could see over here that the fistula tract, understanding which moves like this to the internal opening. So this tract, which is you could see a ultrasonography, and then this could be, yeah, you could see in a different section. This is the entire tract. So normally you need to lay it open or to cut it, you see, from here to here. But this could be the technique could be different where, yeah, we could treat the root cause of this disease and allowing these fundamental principles of wound healing that they, once you remove the main cause, the foreign body, the cellular, the rest of the things body takes care of it, you see. That's the fundamental philosophy of Ayurveda, that if you could take out, you see, you could remove the disease if you take care of the initiative cause of this disease. Here the initiative cause of this disease is the anal gland infection. If we are able to take care of this anal gland infection by Charsutra, we have modified this treatment technique, keeping this basis of Charsutra therapy intact, so you could treat it much better, you see, than completely laying open the entire. This is a newer addition in the conventional treatment technique of our Charsutra therapy. So that what you could see that this tissue primarily innocent tissue that was not diseased. The disease actually starts from here. So if we are able to treat this area, so we need not to cut and lay open the tissues, the innocent tissues. That's the basic thing. So what we do, simply put a small sit-on or a charsutra in between these two places and to lay it open to heal this entire tract, you see. So this technique, what we call now, and technically we call it as an IFTAC, interception of fistula tract and application of a charsutra, is a newer technique to opt this kind of a thing, you see, and that helps to complete it, you see. So what you could see, that's the result is at the end, and what you could see, the finally this heals without leaving any scar and in a very short duration of time, you see. So these techniques supersede the conventional other treatment technique which are available today for management of complex fistula like this. So I feel I'm not going to this. Now the ethics in research, you see, this is one of the aspects what it is quite important, you see, to consider ethics. In our days, you see, ethics, it comes because we carry out research program not for merely that we try to help humanity or try to help people, you see. Mostly we carry out research work because we have a PhD or MD program, we have to submit a thesis, sometimes take a project from the government agencies or some other private agencies. Many a times, you see, we do not bother what this is going to damage a patient or not. There are a lot of such evidences are there. As we know that in Second World War, there are a lot of people in, there were in America, they carried out research in syphilis, though there was a drug available, but they allowed, without any drug, they put the prisoners, you see, without drug, and they try to understand, you see, how the syphilis gradually grow, different stages it goes, and how they die. So look at, you see, the primary objective to carry out a research, not to take care of the humanity. So there are many examples, you see. We do many anti-cancer trials. See, whether the patients are dying due to drug or not, that is not important, but we have to carry out a clinical research to establish the efficacy of a drug, so we carry out. Therefore, a medical ethics came in existence, and now all researches are governed by ethics committee of every academic institution, and now they are rigorously treated. Even today, you can see that in COVID days, that everything has come out, you see, many drugs has come out. They are all immuno booster, you see. So here the issue is not as a ethics, but with how do we promote our business, that was probably the more important issue. But I always believe, you see, these 4 lines described by Sushrut, that a patient, even they do not believe a mother, father, or a friend, they have a doubt, they may have a doubt, but they never doubt doubt a physician or a surgeon. They solely trust on you. Therefore, you treat a patient like your son. If you follow these 4 guidelines, the medical ethics is over. You need not to. So if you see that whatever you are going to do to a patient, is it safe to a patient? Can you do it to your son and daughter? If the answer is yes, the medical ethics is clear. The ethics in research is also clear. But now, these in earlier days, they are up to people. They are authoritative people. But now we are more biased. Therefore, there is an ethics committee for carrying out research work in every. Finally, there are many more research areas. I'm not going to, it is quite difficult to talk these areas, like there is suturing material. Then there are research in research areas in Anusastra karmas, minimal invasive procedures. For example, look at use of maggots in the treatment of tumor has been described by Sushrut. The same technique is being now used for debridement of wound. Now it's FDA approved treatment protocol. So this kind of a treatment protocol has been opted from our own text and classics are being used, but the credit never goes to Sushrut or our Ayurveda. So therefore, we need to relook our own text. Other thing is this, the literary research is one such area. Probably we need to take care of these areas, and hardly there are people who could do literary research. Because literary research, in one hand, you need a person who is quite knowledgeable in literature, Sanskrit literature. On the other hand, he should be equally a trained or a practicing surgeon who could understand the lines between the, the matter between the lines, the thought between the lines. It is not the sloka.
Excuse me, sir.
Yes.
Sir, can you please conclude in 2 minutes?
Within 2 minutes. Within a minute.
Yeah.
It is the line between the slokas are more important. So, which could really realized by the practicing surgeons, you see. Therefore, the literary research, there are only few people are now left for carrying out this kind of a research. Now, these are some of the areas where we could take research in Shalyatantra, literary, historical aspect, as I told you, the biomedical engineering, designing instruments and all these things, surgical techniques. I have given some examples, new drug and development. There are a lot of things you could do to see fundamental principles and conceptual research. So these are some of the areas where you could do our research work. And finally, any research work, the basic objective, I found out this sloka of the Gita, where we say that asatoma sadgamaya tamasoma jyotirgamaya. So this basically that a research is to take to what's ignorance to the knowledge. That's probably the idea of a basic of a understanding of a research work. But now we have deviated from this idea. So I really thank you all for listening me and giving me an opportunity to share my experiences and this research areas in Ayurveda. Thank you all. Thank you very much.
Thank you very much, sir, for elaborately sharing your experiences. And it's indeed your experience speaks louder than the words. Hope all the delegates have got a clear lesson about various areas of research in Shalyatantra. And there are a few questions, sir. If you take, sir. Sir, Dr. Naveen Kumar has asked a question. What are the areas of Shalya research in COVID patients?
In
COVID-19 patients.
Could you please repeat it again?
What are the research that can be conducted, research related to Shalya in COVID-19 patients?
In COVID-related patients. Research areas in Shalyatantra in COVID-related patients. Is it so?
Yes, sir. Yes, sir.
I feel there is some problem in connectivity, you see. Well, to be honestly speaking,
research scope of in COVID-19 cases specifically.
Well, you see, now, to be honestly speaking, you see that there are a lot of ongoing projects which are going on and COVID management. The COVID management is a aspect, and surgical research is a different, of course, you see there are surgical conditions may occur in patients, those who are suffering from COVID. To be honestly speaking, you see that my personal experiences are quite limited in surgical cases, those who are suffering from surgical problems and having COVID, you see, in these patients, because during last last so many months, because of due to my own personal problems, you see, I couldn't have any such surgical procedures or take cases in COVID period. Maybe others can reply these on questions more beautifully. You see, I may not be able to very rightly answer these questions.
Thank you, sir. So there is one more question from Dr. Guru. Suggest some research ideas regarding post-operative pain management.
There are a good number of works being conducted in our department, by the Department of Sangyarana. They tried different procedures that includes drugs, that includes some other procedures. They includes medical management. They includes, you see, management of Agni Karma also. They also did some sort of a acupressure treatment in cases of pain management. But pain is a very complex situation, and mostly this acute pain management, so far this managing a acute pain, right now the drugs which are available to us are opium or opioid alkaloids. They are the most important drugs are being used today. But unfortunately, we do not have right now in our hand a drug which could take care of acute pain rather than a moderate or low degree of pain. You see, they can be managed with many principles. Well, I see there are several situations where pain management is not only related to some drugs also. Certain procedures are also useful to reduce pain.
Thank you so much, sir. There are many other questions with time constraint. I would really thank you for the time given and the knowledge shared, experiences shared. Now I request Dr. Gopi Krishna, Professor and HOD, Department of Shalyatantra, SDMCH, for his remarks on the occasion. Over to you, Dr. Gopi Krishna.
Hello, Dr. Gopi Krishna. Very nice to see you in a period of time. Thank you.
Yes, sir.
Thank you, Doctor. Yeah.
Good afternoon, sir. Respected, most respected Sahu, sir, the most learned stalwart in the field of Shalyatantra. I would like to thank you for your sharing your experience regarding the various research areas in which a student should take in their post-graduation. Thank you so much, sir. And just to highlight some of the areas where in the field of Ayurveda, SDM College of Ayurveda Hassan is conducting. In the field of Jalauka Charna, in case of Dushtarvanas, as sir rightly pointed out that in sir has developed a biodegradable patch called as Panchvalaka Kvata, So students, that is one thought process generating, because sir has done it such a wonderful job. So I would like our young minds to work on Tifla Kvata patch or Manchester Kvata patch, okay, to see whether it is a Varna Ropanga or Varna Shodhana. So sir has given us a very good idea, and he has told regarding Fogarty's catheter. So some of us should try to bring out a particular fistuloscope where we can easily identify the tract along with transrectal ultrasonography. So, sir, and in Jalauka Charna, where in case of Dushtavarna was one of the area, and in case of periorbital lesions where there is loose areolar tissue, Jalauka is the one which will be able to take out the excess amount of inflammation exudates from the surgical site. So there are other particular areas like Arshas, Bagandhara, Ashmari, thromboiditis obliterans, TAO. So my dear students, please look into these particular areas in Shalyatantra. And I would like to wholeheartedly thank ManoranjanSahu sir for his vast knowledge, clinical experience as an excellent surgeon. Thank you so much, sir, for sharing your 35 years of your knowledge, experience to all of us. And from SDM College of Ayurveda in Hassan and our team, we wholeheartedly, once again, thank you very much for being there with us and sharing your vast knowledge. Thank you very much, sir. Over to you, Girish, sir.
Thank you,
Thank you,
Doctor.
sir.
Thank you, Gopi.
Thank you, sir. Thank you very much.
Thank you, sir. Sir, again, I would like to heartily thank Professor ManoranjanSahu sir for being for accepting our invitation to share your knowledge and enlightening us. And on the behalf of management, Principal Dr. Prasanna Rao sir, and all the faculty members and all the postgraduates, all the delegates, I again thank you, sir, Sahu sir. At the same time, I would like to thank Dr. Gopi Krishna. Despite his personal problems, he has come online and he has made remarks. I thank one and all for your active participation, asking question and interaction. Sir, with your kind permission, we shall move on to the next session, sir.
Thank you. Thank you.
Thank you, sir.
Sure.
Thank you, sir. Thank you, sir. Next, I would next I would call upon Mamatha K V madam to the session. Very good afternoon, madam. Good afternoon, madam.
Good afternoon, Girish. Good afternoon.
Thank you, madam. You are most welcome to the session.
Thank you. Am I audible?
Yeah, you are audible, madam. With your kind permission, I would like to introduce to the audience, though you don't need a formal introduction, but as a custom, I would like to introduce you to the audience or present over here. Dr. Mamatha K V, Principal, SDM College of Ayurveda and Hospital, Hassan, is also the Professor and HOD of Professor and HOD of Prasutitantra&Striroga. Madam has secured gold medal in BMS from Karnataka University, Dharwad. She has served as medical superintendent at SDM Ayurveda College and Hospital, Udupi, for more than 4 years. She has served as member of a syllabus committee of CCIM, New Delhi. She is a course writer and editor of Ayush nursing courses of IGNO, Indira Gandhi Open University. She is associated with many NGOs as a resource person in working with adolescent education, women's health and delivered more than 250 talks on these areas of, for the general public. She is a vivid researcher and she has many publications. She has completed a study on Garbhini Rasayana funded by Rajiv Gandhi University of Health Sciences, awarded with a CESEM project with more than 60 lakh funding from Vision Group of VGST of Government of Karnataka. She has completed one more project on PCOS from Rajiv Gandhi University of Health Sciences with more than 15 lakhs funding. She is part of an international project on cervical cancer running in 13 countries as a co-investigator in association with Karnataka Medical College, KMC Manipal. So with this brief introduction, I would request, I would welcome Mamatha Madam to the last session of the last session of the international webinar on synopsis writing and synopsis and scientific writing. Please take over the session, Madam. You are welcome.
Thank you, Girish, for handing over me the session. Good afternoon, all the viewers. Probably by continuous bombardment since three days now, you might be bored to some extent. I hope I will not bore you further. Right? Moving on to my slides. Research areas in Striroga and Prasutitantra. This is the topic what we are going to discuss today. When you join to PG, you will be joining post-graduation course as a fresh postgraduate student. You will not be having, right then, your goal will be to complete a post-graduation. That is your goal. But as soon as you enter into your post-graduation course, I believe you have to make your vision clear, what you want to, what you are going to become, right? Unless and until you search your strength, unless and until you establish your strength, unless and until you are sure that what you want to be, you cannot be very much focused in your studies, right? So when you are joining as a postgraduate student in any department, you will be having three options in your hand open. One is becoming an academician. Whether you will get an opportunity to be that is different matter. You may get a private college, you may get a government college, or you may be teaching at your own circle, whatever it is. But you have an option of becoming an academician. That is one option. You have an option of selecting an area for your practice, becoming a clinician. Maybe you will run a nursing home along with that, or you will be associated with some nursing home holders and you will be joining there as a doctor or consultant. That is the second area what is opening with the post-graduation with you. And the third area is spending your life as a researcher in the core areas wherever you need to work upon or wherever you want to dedicate your life upon, right? So these three areas demand different strengths in you. For example, if you are going to become an academician, you have to be very strong in conveying the things what you are knowing, right? So you have to strengthen your teaching skills during your post-graduation period. At the same time, an academician, there are, if you can, as you can see in the picture, there are some overlapping areas. Of course, an academician should be a clinician as well as a researcher, but his major area would be, or focus would be, to become an academician, to train the younger generation, to train the next generation, in fact. So any flaws in his training will not only affect him, will also have an impact on the future generation, right? Same way, clinician, he has to give justice to his patients. At the same time, he may be teaching something to his patient party, or the colleagues, or the nurses, subordinates, etc. That is a part of academics will be there, but his major area will be to diagnose, to treat the disease, etc. Whereas a researcher needs an intense and keen observation, intense and keen dedication for the area of the research. So you have to focus what is your strength. You have to dig out what is your strength and explore yourself to find out where you want to go, and you have to sharpen your armamentarium, or you have to furnish your armamentarium with the tools whatever is required for your for reaching your goal. That is the first thing I believe what you have to do. Then coming to the
Excuse me,
research
madam.
areas as some.
Excuse me, madam. Have you shared your slides or yet to? Your slides are not visible, madam.
Slides are not visible.
No problem, madam. Please share it.
Oh, I already okay.
No problem, madam. The speech is conveying everything, so not an issue, madam. Please share the.
Yes, not selecting. I'm trying to share, Girish, but it is not opening the option, like share is not taking.
Just a minute, madam. I'll just a minute.
Enable, probably you have to enable.
Just a minute. Now you please try again, madam.
Okay, I'm trying. Now, could you?
It's not. Can you email it to
Just
madam, or so that we can display it from our end?
yeah, well.
Dr. Suhas Shetty at gmail.com.
One second.
Participants, kindly bear with us for a minute. Delegates, we regret the inconvenience caused. Madam will be joining very shortly. Please bear with us. Dear delegates, Madam will be joining very shortly. Okay. Madam, please. Madam, please start, madam. Please unmute, madam.
Slide share.
Please go ahead.
ಕಾಣ್ತಾ ಇದೆಯಾ? ಹಲೋ.
Yes, madam. Is it visible, madam?
ಕಾಣ್ತಾ ಇದೆಯಾ ನಿಮ್ಗೆ?
Just a minute, madam. I'll share it again. Is it visible, madam? Hello. Is it visible now?
Stop sharing. Yes, sir.
Madam, is it visible now?
Yes.
Yes, now it is visible.
Sorry, I'll share it again. Madam, please go ahead.
Unmute. Unmute.
Please unmute. Yes, please go ahead, madam.
Did you unmute me?
Madam, please unmute and you can go ahead, madam. The slides are visible.
Is my voice audible, Girish?
Yes, madam. Your voice is audible, but it is
Here
echoing.
it is mute for me. Next slide, please.
Please go ahead, madam.
Next slide, Girish.
ಮೇಡಮ್ ಇನ್ನೊಂದು ಮೊಬೈಲ್ ಅಲ್ಲಿ ಲಾಗಿನ್ ಆಗಿದ್ದೀರಾ ಮೇಡಮ್ ನೀವು?
ಇಲ್ಲ 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 ಇಲ್ಲ
Madam, please go ahead. Now it's fine, madam. I'll share the slides. ನಿಮ್ಮ ಇದರಿಂದನೇ ಎಕೋ ಆಗ್ತಾ ಇದೆ. From your mobile only it is getting echoed.
Next slide, please.
getting echoed from your end. Can you log in with the mobile, madam, if possible?
Okay, fine. I will. Hello.
Madam, please, you can go ahead, madam. Sir. Mamatha madam, you can unmute and start the presentation, madam. Madam, madam, you can unmute and start the presentation. Huh? Okay.
Now it is unmuted.
Yes, madam. Please go ahead, madam. Sorry, madam, for all the inconvenience.
It was coming as host as unmuted. You're not allowing you to
Yeah,
mute.
because I had logged out. So
Okay, fine.
you can start, madam. Sorry, madam.
I'm sorry, audience, for all the disturbances, what it has created. So now moving on to our slides again. So we were discussing about there are 3 areas in your career which you have to opt for or which you have to choose once you join to post-graduation course. One is you are becoming an academician. Another is you are becoming a clinician. And the third area is you are dedicating your life to be a researcher, right? So accordingly, you have to collect or you have to sharpen your tools, or you can say, I can say the skills of yourselves or whatever the talents you have to suit yourself best to these areas, right? So this is one important area what you have to understand when you join a post-graduation course. So you can suit everywhere, but when your focus is only to one direction, probably you can give the maximum whatever you have. So that is why, though you have to adjust, sometimes life is nothing but adjustment, but if you have an aim, your targets will be more clearer and you can reach your target more aptly and more precisely. Now focusing back on research areas. So as sir discussed with Sahu sir, there are several areas for our research. So basic concepts of Striroga and Prasutitantra, where you can focus on yourself. If we see the very starting topic or very beginning topic, that is Shuddhartha Valakshana. It's a very, very unique concept discussed by our acharyas. So far, it's not very clear. It's not established. There is no scientific evidences to say that what makes Artava a Shuddhartava and how it gets converted into Dushtartava. What are the chemical compositions? What are the biochemical changes? What are the ingredients or contents of this menstrual blood which turns it to be an Dushtartava? So all these questions are still remaining as a question. Though in past few years, there are some studies which are being carried out on proteomic theory of menstrual blood. They say there are more than 5,000 proteomes in the menstrual blood, and depending on the combination, or you can say the collection, or you can say presence of these different proteomes, there will be change in color, consistency, smell, all the factors or the things whatever have been explained in Ayurveda is we can prove that it is nothing but a scientific concept which is yet unexplored, and I believe waiting for some good Ayurvedic scholar to unwrap itself the hidden truth in this particular area. Whenever we are researching on a particular area, we must be very clear that our research should be beneficial to some population, some community, or some science, or somewhere. It should be beneficial. It should not be a research which is carried out with a lot of efforts, lot of money, or whatever it is, and covered in a page or paper or book, and it is to be thrown into the library or somewhere. So what is the purpose of researching on this? If you raise this question, I would say just by analyzing the menstrual blood, instead of going for complex examinations or invasive diagnostic techniques, just collect a bit of menstrual blood. With the invention of menstrual cups, I don't think now collection of menstrual blood is also a problem. So when you subject this menstrual blood for a certain set of examinations, as you do urine pregnancy test now, so similar kits, if we can derive as a startup programs or something, so for the presence of identification of this particular factor in the menstrual blood, or this particular thing in the menstrual blood, maybe simple starting from pH 2, so many complicated constituents, you must be in a position, or we can be in a position to identify different diseases of genital tract. So this particular vast area in gynecology is yet unexplored, and it is waiting and waiting, because since 20 years almost, I am telling my students that this is the area which is unexplored. You can be a researcher in this area. Yes, you may ask why you have not tried. I am a clinician. I have not. I'm an academician. I have not dedicated myself as a researcher. So those who want to be a researcher, probably you can suit best, or you can select this area. And you can also search for some biomarkers, so which precisely tells you about the disease conditions. Apart from this, now even they say that there are a lot of stem cells present in the menstrual blood. Right now we are searching in the cord blood, and there is a huge business going on to protect and save the stem cells from the umbilical cord and placenta. If the menstrual blood can provide the stem cells, then probably this can also be a revolution in the treatment of several diseases, and even cytokine theory in the menstrual blood. So many things are going on. So next slide, please. Coming to Yoni Vyapat and Artha Vyapat concepts, then there is, for all the diseases, whatever said in Ayurveda, gynecology, particularly Striroga, we have one line, or at the most 2-line or 3-4 line descriptions. There is no clear-cut samprapti. There is no very elaborate descriptions that how dosha dushya sammochana takes place, to what extent the doshyas will be involved in this pathology. So every disease, if somebody can, maybe along with the Roga Nidana department, in collaboration with the Roga Nidana department, if we can draw a samprapti of each and every disease, probably it would be a very good contribution to the literatures of Ayurveda, Striroga and Prasutitantra. Then Amshamsha Kalpana of the doshas, wherever in a particular given disease, what dosha is involved, with which gunas it is involved, to what extent it is involved. If we can have a clear-cut description of this particular Amshamsha Kalpana of the doshas in individual diseases of Striroga, then it can help us in the selection of drug. What kind of drug is to be selected? If Ruksha Gunataha Vatavrati is there, probably a Snigdha Vatahara Snigdha drug will be helpful. Similarly, Pitta increased with the Drava Guna. So what would be the ideal drug of choice in this area? So from these, like deriving pathogenesis of the disease, then correlating the underlying pathology with whatever said in Ayurvedic Samprapti, that is also pending. Then sometimes we can say, just example, if I have to give you an example, Asradhara. There are varieties, 4 varieties of Asradhara, Vataja, Pittaja, Kafaja, and Sanipataja described in the classics. If you analyze the Samprapti of Asradhara, they say that Rajovaha Siraktaha Raktam Pramanataha Utkramya. So Pramanataha Utkramana of Rakta is taking place and it is getting Ashraya in the Rajovaha Sira, Garbhashayagata Rajovaha Siraha. So indirectly it is pointing towards the congestion in the pelvic vessels. So if there is congestion in the pelvic vessels, that is the common Samprapti or that is the common pathology you see in all gynecological conditions where there will be excessive bleeding. So the description of Asradhara, we can say it's indirectly saying about the abnormal uterine bleeding, all varieties of abnormal uterine bleeding. If so, then Vataja type of Asradhara particularly may be having some underlying pathologies. So what kind of pathology gives rise to Vataja Arthava Lakshanas, Vataja Asradhara Lakshanas? What kind of underlying pathology will give rise to Pittaja Asradhara Lakshanas? Like this also, you can summarize, you can do an observational study, or there are a lot of studies what you can carry out, and similarly, the research. So also you can just observational study may suffice in these cases. So this year we have carried out a study on Asradhara on the basis of Vidhi Shoniti Adhyaya. All the Nidanas of Asradhara are compiled and we have analyzed in how many patients we could get Rakta Dushtikara Nidanas like this. So what are the giving causes which give rise to a particular disease and through what mechanism they are giving rise to the Samprapti of that particular disease. So all these, if we can elaborate, that helps the younger generation or next generation to understand the disease in a better way instead of reading a single worded shloka like one Dhyana Startava Katita. This is a Samprapti or this is a Lakshana or this is the Purva Rupa or this is everything of one particular Yoni Vyapats. So one Dhyana Startava Katita, if Acharyas were very Tikshna Buddhi, so one word description might be sufficient for them to understand. For us, Manda Buddhi Shishyas, probably we need to elaborate it. If somebody can elaborate, somebody can dedicate few days or few months or few years to elaborate this particular thing, it will be a great help for the future generation. Next, coming to standardization of Sthanika Chikitsa. So conceptually we say, or practically also we do lot of Sthanika Chikitsas. Everywhere Sthanika Chikitsas are practiced. Everywhere they speak about Sthanika Chikitsa. But if a core question is asked, whether your Sthanika Chikitsas are standardized, the answer would be mere no. If you go to 100 places in India where Sthanika Chikitsa are practiced, 100 people will have their own 100 methodology of implementing this or performing this Sthanika Chikitsa. It's not a problem that they do it in their own method, but in all the methods or in all the ways, there would be an ideal method. If you give it in that particular way, the intention or the purpose of giving would be served in the best possible way. So in what way it is, if we have to analyze, it might be in terms of dose. For example, Kashaya, in what dose for the Shalana, if it is given, it will be optimally used or it will be giving us optimal benefits. So if you are giving douche or if you are performing Shalana, with what temperature of the liquid you have to perform that Shalana, with what temperature the absorption would be ideal or very high, the question. Then what form of drug formulation would be more ideal, whether a Sukoshna, Kashaya, or a Swarasa, or what formulation of drug is absorbed by the vaginal mucosa in a better way. That is also a question. What should be the drug concentration? Initially, we were using 500 ml of a particular Kashaya to give vaginal douches. Later, I thought, why should we waste 500 ml of Kashaya? Instead of that, we prepared a very concentrated Kashaya of 200 ml with the same drug, and we started giving a slower douche in the same way, but we started giving it a very slow pace rate. So then my results were better than the dilute Kashaya which I was giving with the 500 ml. Though the quantity was more, the results were better with a more concentrated and limited quantity of the Kashaya when I administered. Right? Time and drug contact time. Definitely vaginal mucosa becomes permeable for the drugs at a particular period. So that optimum time, it is already standardized by the modern doctors. But from Ayurvedic point of view, maybe Kashaya penetration, maybe Kalka penetration, maybe Varti penetration. So all these things, we need not blindly accept whatever they said. Transmembranous route absorption studies can also be carried out with our own drugs, our own modality of administration, and we can standardize what should be the time of a cycle when the drug has to be given. At the same time, to what period the drug and the vaginal mucosa should be in contact. So drug contact time is a major factor which decides the absorption rate. Right? So this can also be thought about, and mode and extent of absorption of the drug, whether the drug is hydrophilic, whether it is lipophilic, to in what form the given drug is absorbed, whether we need to add some, in the Vartis, for example, we keep a layer of Sneha in order to lessen the irritation caused by the Vartis. So whether it is needed. So all these questions are still remaining as a question. Then impact of Purva Karma. Of course, blindly we say Purva Karma enhances the circulation to the local area. So with this enhancement of circulation, drug absorption would be better. But to what extent absorption would be better? Is it increased by 20%, 40%, 60%? So all these questions are still questions. And if anybody, it's not a task of one person, but one person with one particular smaller area in Vata Prakriti or Vataja disorders. This should be the dose, this should be the temperature, this should be the formulation. Starting from a simple, like Aliluseve it is, if we start thinking about standardization, probably that would be a great contribution of a research scholar to the basic concepts of Ayurveda. Then coming to Striroga, Prasutitantra area. So Garbhadana Samskara. So Garbhadana Samskara, it's said a lot about it, right? How many Shodhanas are required? Even in a healthy person, whether Pravara Shuddhi is required, Madhyama Shuddhi is required, Avar Shuddhi is required, whether all the Samshodhanas are to be given, if Samshodhanas are given, because Madhyama Mradu Koshta patient in Arthavadushti, it is said that Mradu Koshti patient, if Tikshna Aushadhis are given, or if Pravara Shuddhi takes place, probably without dosha, if we give, that is giving rise to complications of Arthavadushti. So same way, Garbhadana Samskara, it is said, but in what way it has to be practically performed, we have never thought about. We are here and there are some studies, but we are not in a position to say confidently and to draw a conclusion, concrete conclusion regarding this is required and this should be done. Right? Then coming to mantra, several mantra chikitsas are said. We are, of course, most of the times we are not practicing, but there are people who claim that Prasava mantra, one of my own students, she says that Prasava mantra is very good in delivering patients normally. Madam, you tried, she was telling me. So whether mantra siddhi is to be done or not, if I get the mantra, conquer, or I can say mantra should be, you have to do the siddhi of those mantras by chanting so many times and in such situation, such things are said. whether mantra chikitsa, if one has to adopt, whether he has to do the siddhi of that mantra, we don't know. Then there are some yantra, Ubhayatrimshat yantra is said in Ayurveda for Sukha Prasava. Whether that yantra is, if just written on a paper, is it enough? Or it should be written on a metallic sheet, whether it should be kept below the bed of the patient, or whether it should be kept in the labor theater, whether she has to see that, whether it is just diverting her mind to the yantra to ease the labor pain. All these questions, in spite of several years of practice, many of us, even practitioners, we are, or the, teachers, we are also having this as a question. Probably we can think over these areas and conclusively we can derive some opinion what way it is to be done. Varanabandha, Putra Kamesh, Diyagas, all these come under the same category. Then coming to one of the very subtle area which has not been explored is Garbha Chalana Chikitsa. So Masana Masika Garbha Vradhi or Garbhini Aushadhis are said. At the same time, Garbhachalana Chikitsa is also described in Ayurveda, but Garbhachalana is given to prevent abortions or to prevent premature labor. So wherever it is, unless and until you carry out some standardizations, or you can say some pilot studies on this, there will be, if you give them confidently, there will be some ethical issues. By chance, if the patient starts getting aborted, or process of abortion or premature labor continues in spite of your Garbhachalana Chikitsa, ethical questions will arise that who has given you the right to give this? Probably if you had given Duodilan, or if you had sent her to modern doctors, probably you would have saved it. Right? So regarding this, to what extent these drugs are having potency or capacity to prevent abortion or to prevent labor, all these things will remain as a question. As a matter of fact, if I have to say, we have carried out a study in the 8th month Garbha Chalana Chikitsa, Pippali and Lodra Pippali in Godugdha and Gogratha, I think, combination around 5-6 years back. And that has given me good results. Encouraging, I won't say in all the patients we did not give Duodilan, we could avoid the use of Duodilan, but relatively it had some action. So it is an encouraging factor that probably every month, of course, we have conducted some pilot studies on Masana Masika Garbha Chalana Chikitsa also, but Garbhini Avastha and progress of labor is physiological. Because of the drug, definitely I could prevent it. I cannot claim it. Maybe single digit trials, number of patients in whom we tried this Garbha Chalana Chikitsa is in double digit, not in single digit. In spite of that, I am not in a very conclusive state to say that in a position to say conclusively that it is definitely working. So such areas we can explore, we can do some researches. Then Rasayana Kalpas of Kashyapa, a lot of people are using Chatapushpa, some are using Chatavari also, and some are using Lashuna also. In what all kind, it is said that it is like a nectar. It works in amenorrhea, it works in excessive bleeding, scanty bleeding, it works in infertility. Its scope of utility is very vast, but we have not precisely said in what dose if it is used, in what conditions if it is used, with what anupam if it is used. It is useful in dysmenorrhea, it is useful in amenorrhea, it is useful in dysfunctional uterine, it is useful in infertility. So unless we standardize, we cannot say this drug is helpful in all the conditions, right? Maybe sometimes it works also. I do not say it is not working, but we should have some rationality, we should have some data, we should have some clinical trials to say that one particular drug works in all conditions, at all circumstances, and gives us result. Then concept of Jataharini. Recently we had a CME. In that CME, when I thought of giving Jataharini to several speakers, most of them said, oh, Jataharini, no. So that is the area about Jataharini. So it is so complex that no one could understand what is this Jataharini, whether it is a psychological condition, whether it is a condition requiring only Daiva Vyapashaya Chikitsa, whether it is a condition like Krimi Samsarga or like Corona, something, right? Upasargas, we don't know, because its Jataharini also is said like Janapado Dvamsavyadis, it spreads by using one's garland, this dress, etc., etc., and not following the code and conduct. All these rules and regulations have been put in acquiring this Jataharini. So is it something transmissible disease like microbial disease or something? It's not very clear. Then the very core area of gynecology, if we think about it, is Vegadharana. It's said as a Nidana for almost all gynecological conditions, starting from simple Udavarta Yoni Vyapat to major pathogenesis, right? What is the impact of this Vegadharana on genitourinary system? Whether it works on nervous tissue, whether it is working on muscular tissue, whether it is working through some blood vessels, how it acts, how it works, and on what plexus, neural plexus it is working, it's not standardized. We have not studied. Of course, we see clinical results. We see that if the patient does Vegadharana, she will develop Udavarta. It may further aggravate into or progress into the Vatiki Yoni. And if she leaves this Vegadharana, then next cycle or next few cycles will be painless and Udavarta will get relieved. And again, if she continues to have the Nidana, Sevana, further the disease progresses. So all these things, it is a very important area to ensure that this Vegadharana, how it acts on the pathologies of genitourinary system. Similarly, the studies on Mudagarbha, we are all screening, the public are screening, the government is raising alarms regarding why the incidences of cesarean section is increasing. Is there any way to cut down the incidences of cesarean section? So why can't we look into Mudagarbha Samprapti? Why can't we look into Nidanas of Mudagarbha? Why can't we think by adopting Nidana Parivarjana of Mudagarbha and taking a pregnancy in a smooth course where Apanavata Dushti will not take place, thus resulting into Mudagarbhas, even a retrospective study where hundreds or 500 of cesarean patients who underwent cesarean section, we have to analyze what was the Nidana in them to bring about this kind of Mudagarbha. So maybe we have to classify Mudagarbhas like Ashtagatis or Trividagatis or Chaturvida types of Mudagarbha. Then which particular Nidana has given rise to this particular Mudagarbha and what was the indication in this particular patient for the occurrence of cesarean section. This will also be a voluminous study and definitely it can give us some inputs regarding if the patient is indulging in this particular Nidana, probably she will not have normal delivery. So this would be a very good area for any researcher to research on it because there is no scarcity of subjects in this particular area. In a hospital, if you are having 100 patients, then around 60 to 70% will be of cesarean section and plenty of subjects you can get in this. Then regarding Artava Dushti Lakshanas, Shuddhartava Lakshanas, to some extent, we are evaluating in the clinics. But Stanya Pariksha, very, very few places, at very, very few centers, probably we are opting for Stanya Pariksha. Even if we are doing Stanya Pariksha, that we are doing just for the sake of demonstrating it to the students. But the application of Stanya Pariksha, if we do, probably it will save a lot of our children from getting a lot of antibiotics, newborns from getting antibiotics or several unnecessary medications in the very early days of life, definitely, which will have some impact on their future health status. Same way, Shukra Pariksha, maybe in Jamnagar it was there, but I don't see anybody practicing this Shukra Pariksha as per the Ayurvedic norms. So Shukra Dushti Lakshanas, evaluating Shukra Dushti Lakshanas in a given sample of semen, if that is also incorporated, probably better results we can expect. And as
Artava Dushti Lakshanas we treat sometimes. And same way, Shukra Dushti Lakshanas also, if we can treat, probably we can expect better results in the area of infertility. Then moving on to a lot of basic areas are there which are untouched and which a researcher can explore. Then coming to the clinical practice, in clinical practice, research areas are impact of Shodhana in preconceptional care, as I discussed. So what happens, that is why it has to be done, how many times it has to be done in the concept. So if one particular Shodhana is done, Virechana is done, what is its impact on labor? What is its impact on conception? What is its impact on the course of pregnancy, whether the patient had more emesis, whether the patient had no emesis, whether the patient had only normal nausea or vomiting, or whether the patient landed up with the anemia of pregnancy, whether she had any other complications of pregnancy, whether she ended up with abortions or anembryonic gestation or IUGR, etc. All these are still questions. There are scattered studies here and there, but as I discussed, there was no one solid conclusion about the impact of Shodhana Chikitsa in the preconceptional period on outcome of pregnancy. If this is about normal pregnancy, a patient is coming to you with a complaint of bad obstetric history. There is history of previous 3-4 abortions, or there is a history of previous anomalous baby. There is a history of baby with IUGR. There is a baby with a conjoined, there is a history of conjoined twins, or there is a history of some other repeated cardiac condition in the baby because of which, baby, because of which repeatedly she underwent about termination of the medical termination of pregnancy. What is the impact of our preconceptional care in this area? Then, if the patient is having some previous history of pregnancy complications like gestational diabetes mellitus, pregnancy-induced hypertension, hypothyroidism during pregnancy, hyperthyroidism during pregnancy, preeclamptic toxemias of pregnancy, excessive edema during pregnancy, oligohydramnios, polyhydramnios. In her previous pregnancy, she had all these complications. So after Shodhana, during this pregnancy, whether she is having all those placenta previa, that can also be taken as, right? So these complications, whether they are recurring pregnancy or the Shodhana, what we have given, corrected the Apanvata, corrected the symbiosis of the doshas and or balance of the doshas in the body. So whatever has occurred during this pregnancy, it is in a smooth, balanced, normal, very much physiological pregnancy she had. That can also be a conclusive remark of this study. Then impact on labor, how the labor took place with this preparation. Whether it was a normal labor, whether it is a cesarean section, what is the baby weight, what is the baby condition at the time of labor, follow-up of baby's milestones with the pre-conceptional care. A few clinical researches, clinical trials, what we have conducted, the parents are telling that these babies are relatively more faster and more wiser and more active than other children in their home, those who have taken preconceptional care, one area. And it is also a common comment on us, on our SDM Udupi people. The parents are telling that SDM makkalu tumba tante. That means SDM children are very, I can say, active rather, it's not in a good sense they use. They're very mischievous. So mischievous in the sense, if a baby is very active, that shows that it is very talented, or anyway, it's not dull, right? So maybe it's having the rasayanas, what we give during pregnancy, the antenatal care, what we extend to the mothers, the gratas, what we give to the pregnant ladies, probably are working in this area to enhance the IQ of the babies, or the capacity of the babies, or the intelligence of the babies, that we cannot ignore at all, right? So coming to antenatal area again, in continuation with the preconceptional care, if the antenatal care according to Ayurveda, if it is done, what are the outcomes on common ailments, maybe mesis gravidorum, simple shotha, constipation, hemorrhoids, varicose veins, all this, right? Then pregnancy-induced diseases, as we discussed earlier. Then medical conditions in pregnancy. Simple anemia, that can also be taken. Garbhani Paricharya and its implications on fetal immunology. We carried out a study where in 6th and 7th month, we have given some Rasayana Aushadhis to the pregnant mother, and after delivery, we have taken the cord blood analysis. There was a marked difference in the IgG, IgM and IgA levels of the fetus, of the cord blood in the trial group with comparison to control group. That we have given for only 2 months. If the entire period of pregnancy is given with these rasayanas, and if we had done a study, you guess what would be the results. See, we are sitting on the treasure and we don't know on what we have. It is like Karas Chandana Baravahi. That simile, I remember. So we have not conducted an authentic study. I agree, there may be lack of patients in some centers. There may be patients who are reluctant to take bhratas, reluctant to take rasayanas. They ask us for matrakodi, that may be there. Give me tablet, maybe their version. Or there may be some centers, most common problem what we face in this area is immunoglobulin studies are very costly. A PG student cannot afford for these studies, or the patient may not be affording for these studies, or for our sake, immunomodulation or immunoglobulin study is patient has nothing to do with this, right? Patient needs a healthy baby. That is our intention. For our sake, that we want to prove that our science is greater, our science is giving them some benefit, we cannot tax the patient to bear the cost of these investigations. That is the most common practical problem. Probably we can write small projects in this regard. And sometimes if the patient is, not all patients are having all patients are having economical barriers. So those who are willing, those who are good, those who are having good economical status, probably we can think of carrying out these studies. So that probably that may not be of course, if you have a good number of patients, you can try with the PG level only. And for PhD studies, probably this can be taken as an ideal study. But whatever studies we are carrying out on antenatal, I know there are some studies being carried as multicentric studies also. But in the National Protocol of Antenatal Treatment or Family Health and Welfare Department, so far we have not presented ourselves. We are not there with any of our studies wherein we conducted studies with 1,000, 2,000 or 5,000 patients, and we said that this is very useful combination. If you give this combination in this particular, maybe you tell that it is individualized medicine. Yes, you give the criteria, sub-criteria, all parameters or whatever the conditions you apply. In Vata Prakriti, in Jangala Desha, in normally constitutional patient, this particular drug is acting. This hematinic you can give, which increases the patient's hemoglobin level to 1 gram per month. So there is lack of such large studies, large sample sized studies. If we can collect a data from multiple centers conducted simultaneously on the same drugs, probably we will have a lot of database with us, with which we can approach the government to say that this is most ideal hematinic, which will not have any side effects as a iron supplementation would have. And we can also say that this is more congenial to the patient. It is herbal origin, or you can say this will not cause any harm to the patient. So with these evidences, when we generate the evidences for the benefit of the patient, for the proving the benefit of the patient, then probably we can do a lot good in the areas where we are working. Next slide, please. Next slide, please. Next, we are coming to area which I'm most interested, that is labor management and Ayurveda. Probably we have tried not less than 10 drugs in labor management with Ayurveda, but there is no single drug with which we can say that definitely without oxytocin, I can run my labor room. Right? We have tried Dashamula, Vasa, several Erandapaila. There are many more drugs which are unexplored. Probably my meager experience is insufficient to say that this drug is an effective replacement for oxytocin. So the day, I often used to say that the day we can say that we can run labor room without oxytocin, that would be the best day for Ayurvedic obstetricians, right? We need not give any of the injections. Just you give some lepa, you give some dhupa, you give some vatha, you give some swarasa. Of course, I can say around 60 to 70%, rather 80% of my patients can be delivered normally without oxytocin if I am ready to wait. With all normal interventions, preconception, post-conceptional care, pre-labor care, that is 8th and 9th month bastis, if we give, majority of the patients would go for normal labor without oxytocin. That is true. But for the rest, 20 to 30% is the question, because labor itself is physiology. So such studies, all the drugs, wherever it is said that this particular drug is oxytocin, maybe we can also search in folklore areas what our ancestors used to give when there was non-availability of oxytocin. So these areas we need to explore to establish or to overcome the practical problems what we have faced during our establishing years, our practicing years, and at least for the next generation, we can give some nectars on which they can say that with Ayurveda, we are practicing independently without the support of some modern drugs. Then coming to the research areas in Sutika Pakala, maybe post-operative replacement of antibiotics in the post-episiotomy or in the episiotomy wound or in the post-operative wound. All these areas are researches are going on, but I don't see anybody without antibiotics practicing surgeries, even at least prophylaxis. Of course, I know when we PGs were not there in my center, we were giving one single shot of prophylactic antibiotic and we were doing cesarean sections. And in normal deliveries, we were not giving antibiotics. So it is not a single factor that only drug has to see the wound healing, but it is also true that drug is also an important factor. Even though PGs are doing cesarean sections, with antibiotic we are covering, and with the antibiotic we are confident that the wound will not get infected. So any drugs which enhances wound healing, that can also be given a good trial. Probably pharma companies are waiting for your result, and if we can do patent on some drugs that this is the best wound healing, we can claim that with this cesarean wound heals. Even if you do not apply anything, it heals. That is true. But if anything can enhance wound healing, that because many pharma companies, they claim that you apply this bandage to, you apply this clip to, you apply this powder, everything. A lot of pharma companies are based on this area. So probably we can also come out with some things for at least for patient satisfaction that it enhances. Then always we think about quantitativeness of the breast milk. So it's not only the quantity, probably we have to switch over to qualitative analysis of breast milk also. So if breast milk enhances the drug enhances the quality of the breast milk, qualitative analysis of the breast milk can also be thought about with the drugs what we give during Sutika Paricharya. Then the patients who had GDM, who had pelvic inflammation, pregnancy-induced hypertension, we say that within 3 to 5 years, there is a chance of them getting into diabetes mellitus or converting into hypertensives. But I would say that if the Kleda Samprapti, which is the cause for these diseases in the antenatal period, if handled well, Sutika Paricharya itself is a stage of cleansing. It's a Shodhana Kala. So during this Shodhana Kala, if the Shodhana is done perfectly, if the cleansing is done perfectly, probably we can challenge that. Patient will not develop, provided patient has to be a Bishagvashya and she has to adopt the all Sutika Paricharyas. After that also her Dinacharya, Rutucharya, her Vihara should be congenial. as we prescribe to her, and we can challenge that they will not develop as diabetes mellitus or hypertensive patients in future, at least till they are 50 years or 60 years, definitely not at 30s or 40s. So this is the area where we can work about. Then addressing the psychological issues, maybe because of loneliness, maybe because of lot of responsibilities, maybe because of work stress. Many puerperal women are entering into puerperal blues or puerperal psychosis like conditions. So if we can support them with appropriate Sutika Paricharya, probably that would be a great contribution in converting our mothers into healthy mothers, cheerful mothers. Right? Next, moving on to research areas in clinical practice of Striroga. So nowadays we are seeing that children at the age of 11, 12 years, they are getting menarche. Then equally true is patients, those who do not get menarche even at 16 years of age. Sometimes it may be simple, there's no pathology, but when there are no secondary sexual characteristics also, then it becomes a question mark. So in these areas, if we can do something. Developmental problems such as subnormal uterus, hypoplastic uterus, I heard and I saw many cases by giving some Basti Chikitsa, by giving some additional Ayurvedic Rasayana therapy, the subnormal or hypoplastic to some extent, I do not say a streak gonad or a small bulb-like or some descriptions ultrasonologists will give, that it is like a small streak like uterus, a small bud like uterus. So all these may not change, but a subnormal size, a small uterus, which is said as hypoplastic many a times, getting converted into normal uterus, or the lady achieving pregnancy after marriage, those who was, the girls who were not having menstruation regularly, getting regular menstruation after treatment, probably I would not say this is simple chance. So this needs also, the incidences are very less. Probably this would be a combined effort of all the teachers or the postgraduates over a long period of time, probably 25, 50 samples. If we can collect together, that would be a contribution to say that any hypoplastic patient, she need not go only for hormonal supplementation, just for namesake, for the growth of it or some secondary sexual characteristics, and probably her future would be a gray area with respect to reproductivity. Whereas with our treatment, we can definitely say there will be a result. So such gray areas for research where samples are very rare to see in the population can also be with a teamwork. We can collect and we can establish. Then preventing hysterectomies at so many conditions, maybe PID, chronic PID, maybe fibroid uterus, maybe a dysfunctional uterine bleeding, so on, so on. So abnormal, unnecessary sacrifices of the womb definitely can be prevented, because many patients with the endometrial hyperplasia, with the dysfunctional uterine bleeding, when they turn to Ayurvedic medicines, we find them giving, going back with the successful treatment. So such areas we can take up for researchers. Endometrial hyperplasia, like drugs given. So what is the impact of it? Such studies we can definitely take out, because the sample size is not very less in this particular areas. Then maybe as a trial, we can also take up the studies, because fibroid, PCOD, all these, we say that it runs with the genes. So mother had fibroid, sisters or siblings had PCOD, what about the siblings or the daughter? So prevention of fibroid, PCOD is a need of our, how we can prevent them. Of course, apart from lifestyle modification, if we can give some drugs to prevent, or we can give some inducing drugs in the animal experiments, and we can give some combating drugs to neutralize this effect. That can also we can think of. So management of all gynecological small to large yoni kandu, starting from pichila yoni, etc., whatever we see, all those things we can think about. Prolapse management is a rare condition, rare possibility according to modern. First degree, second degree prolapses effectively we can treat, and urogynecology, stress incontinence, urge incontinence, definitely in modern science, except Kegel's exercise, they do not have any solution. Whereas our drugs, our Uttarabasi interventions, all these will have a wonderful result to the patient, and the stress of her life can be converted into the quality, rather in simple words, we can say the quality of life with these complaints can be changed with our approach. So in those areas also, you have a scope to do the researches. Next, moving on to gynecological oncology. So wherever we go for early detection and prevention, definitely its result is better. We as Ayurvedic physicians who are very near to the population, so we can take up so many studies on early detection of cervical cancer, breast cancer, etc. We can give we can identify a population where the family history of BRCA gene is there, family history of breast cancer or cervical cancer is there. In this vulnerable group, we can target and we can advise them with the lifestyle modifications or other studies, other things, Rasayana Dravyas, Shodhana, etc. Then we can see the possibilities of development of this cancer in those patients. So that can be taken as a very potent study. Then reversal of CINA, CIN 1 and 2, they say it is self-reversible, whereas CIN 3 is a progressive condition according to modern. So CIN 3, if we can revert them, CIN definitely, it gives us some time. If we can convince with some pilot studies, with the consent of patient, with the Shodhana and Shamana Aushadhis, if we can revert the CIN, or sometimes we may also use here Lekhana Karmas, or we can also use Kshara therapy, Agni Karma, etc. So together, if we can revert the CIN to normal condition, that would be a great achievement from Ayurveda side. We can think of that. And tumor pathologies, wherever there is a benign tumor, if we can replace it, or we can regress the size of this tumor, maybe we can give them simple symptomatic relief. We can see the literal tumor regression in these patients. And even though the tumor size is not regressed, sometimes the pathogenesis is reversing as evidenced by tumor markers. So that can also be taken up in several benign areas, even in the breast disorders, fibroadenoma breast. We have carried out a study. Initially, we took it as animal experiment. In animal experiment, we got good results. So encouraged by that, we went for clinical trials on fibroadenoma breast. Definitely, we had some encouraging results in this area. So there are some drugs which are said in Granthi or Buddha Chikitsa that we can adopt. Then area of contraception. In Germany, they are using plant-based oral contraceptives. The nation which has the claim or credit of having Ayurveda in its roots, yet we fail to recommend a single herbal or herbo-mineral combination as an at least option for hormonal pills. That is the tragedy of Ayurveda. There are a lot of scattered studies right from the day when I was studying. Before I joined Banaras, there was study on Tanpana Vidanga Pipali that has given this result, that result. Yes, as I said earlier, the results were wrapped up in beautiful covers or beautiful packing, and they have been made a booklet and they have been deposited. That is not brought into practice. So definitely we need to be alert. We need to have some eyes on this area, which we definitely can explore. And if we can explore, if we can come out with some solid results, we can recommend the National Family Planning Board that those who do not, there are many patients who do not want to consume modern pills, right? In such areas, at least, if we can give some of our pills, that would be a great contribution. Then, of course, prevention of menopausal. It was a style few years back, going for HRT, hormonal replacement therapy. Then gradually they got to know about Ayurveda, the drawbacks of hormonal therapy. In spite of that, some are taking it, some are suffering with all these complications. But if we can come out with a particular rasayana which can prevent the aging, established through different markers, maybe biomarkers you can use. You can say that neurotransmitter serotonin level increases. That is why mood elevation is there. Depression goes off. You can say anti-aging property because of free radical scavenging is there. You can say reduction in oxidative stress is taking there. All this, blah, blah, whatever. You can use these markers to establish that this particular rasayana is wonderful in case of menopausal complication. You can classify that hot flushes, this drug. This particular complaint, this drug. So if worldwide, nationwide, a campaign is done that in this particular area, this particular center will work on this. This particular center will take up this research work like that. Or if a particular teacher or an academician reserves or dedicates its life saying that I will work on maybe various drugs for years together. PGs will work on one particular topic, but if a guide is working on that particular area for 10 years, he will be having 10 different studies or 10 studies of different levels, right? So we have to gather, and instead of having some researchers of scattered manner, if we can compile and if we can contribute something significant to the area of practice, that would be ideal according to my view. Then coming to research areas in infertility, it's an open door. Everyone treats infertility. Everyone says this drug is useful, this drug is useful. Everyone asks other that, what is your experience? But is it possible? They say a lot of results that we get wonderful ovulation with this. This year we carried out a study on endometrial receptivity, Ayurvedic Matra Basti, how it improves the endometrial receptivity. So such advanced apple balm, through apple balm scoring, we are proving endometrial receptivity. We are giving Uttara Basti in endometrial receptivity. We say Uttara Basti is working on endometrial receptors and it improves the fertility. So many things we are saying, but is there any center in India which takes up assisted reproductive work with exclusive Ayurveda? If yes, then we can say yes. Ayurveda has a definite role in fertility enhancement. I will say you use the technology, you collect the ovum, you fertilize it outside, you bring it back to the uterus. But the drugs, what they give for ovulation, the drugs, what they give for endometrial preparation, the drugs, what they give for implantation and further growth and continuation of pregnancy, if that can be done through Ayurveda, that is the golden day for Ayurveda. That is my dream, in fact. Now coming to, from clinical areas, research in drug development. What are the areas or scope for drug development in Ayurveda? So there are a lot of researches done on extraoral taste receptors, right? So particularly they say Tikta Rasa receptors are there on the uterine musculature. So if somebody takes up a Tikta Rasa drug, maybe Lodra, right? Like this, maybe Tikta Rasatmaka, what is the impact of this on uterine receptors? Why Lodra is acting as stamina, right? Same way, different drugs may be working at different levels of receptors or different, some may be working on musculature, some may be working on nervous tissue, some may be working on receptors. So various, their target area may be different. How it works? So this, if we can establish, that would be a good contribution. As I discussed earlier, there are a lot of oxytocin drugs, abortifacient drugs. Instead of giving RU486 or mifepristone, misoprostone, if we can give some Shodhana drugs, Shodhana in the sense Lekhana and Bhedana drugs, and if we can establish at 40 days, 50 days, instead of going for that drug, of course, this is a double-edged sword. We have to use it very carefully. But if we can give it through Ayurveda, will it not be ideal? Instead of depending on those harmful chemicals. Then Agar Sangrahaniya Gana Aushadhis, it's they are in Prajastapana Gana. We carried out a PhD level study on this. We found out that there are some progestogenic action of this Prajastapana Ganaushadis. Similarly, Rakta Sangrahaniya, there are some Vedana Stapana, Chonita Stapana Gana, like whether several Ganas are said in Ayurveda, which are applicable in the area of Striroga and Prasutitantra. Their evaluation not only in terms of clinical efficacy, initially pharmacological studies, preclinical studies, all should be carried out, including the teratological studies. If a study is getting completed with these aspects, then only there will be universal acceptance for the drug. So such kind of international standard researches has to be taken up. Maybe in a collective way. 1 year you will do some researches on a simple thing. Next year, the other scholar will add up to its pharmacological. Some scholar will work on clinical. Some scholar will work on teratology. So like this, in a lifespan of a teacher, probably one drug can be evaluated, or evolution of the drug can be done to its maximum extent. So that kind of approach should be there to the research, in my view. Then we are using a lot of drugs in Garbhini Paricharya, but when the international forum asks us, what is the teratological studies carried out on these drugs? Are they safe during pregnancy? Particularly on Rasaushadis, their question would be, but still, even on Rasaushadis, if they say, is there any teratological studies? Our answer is mere no. Shouldn't we think about this area? Then, as I discussed, hemostatic drugs are, there is Lodha, there is Mustha, there is Manjista, there is Ashoka, there is Ushira, there is Chandana, there is Raktabola. So many drugs are there. Are they working in the same way? Whether their mechanism of action is same? I don't think it is same. How it is acting? That we need to establish. Whether they are shrinking the capillaries, whether they are stabilizing the blood vessels, whether they are inducing contraction through receptor-mediated channel, whether they are simply contracting the uterus, whether they are neurologically stimulating the uterus to get contracted. So whether they are stimulating the hormonal mechanism to release more progesterone or to contract the uterus. All this is still a question. We are prescribing the drug, but we don't know how it acts in the body. Maybe we are in a position to answer it is Vata Hara, Pitta Hara. I agree. But at the same time, if we can get result for this, will it not be more nice? Act in quick response in facing new challenges. For example, now Corona. It's evident every one of us say that we give good results in Corona treatment, but none of us are coming out with confidently, or of course government is also having not a very favorable hand at us to say that you come up, we will help you to establish your science. So that is there. We should not discuss on that. But anything, it's Corona is something small thing. Apart from that, if we can take up the challenge in the with the data, if we can prove. For example, in the initial days of Corona, we have studied on 10 Garbhini patients. We said that 10 Garbhini patients did not have any problem. So such data, if we can provide simply on our words, nobody will believe. If we can challenge, we can, with the challenging, if we can take the data, that is establishment. Exploration of nasal route, Vinasya is said to be a very good route. Again, there are scattered studies, how it acts, what is the modus operandi of this, HPO axis, how it stimulates. I do not say there is a lot of work happen on this, but a monograph on this. You can compile, even the studies can be carried out to compile some things which have a vast research, but it is not established yet. You can take up such compilation work also. Once in a while, you can go compile all the studies. Then you say that all these studies established that this has a role. So probably this can be recommended. Such conclusions also you can give in the area of gynecological practice. Then one more treasure, what we have semi-explored, I can say. I don't say it is not explored. Folklore treasure. There are a lot of folklore practitioners who say that by giving this drug, she conceived. By giving this drug, we surprisingly sometimes they will have stopping of bleeding. Bleeding which was taking place since many days, it will suddenly stop with a kashaya, one kashaya. Whereas we give one powder, one tablet, one kashaya, whatnot. So they stop it with one kashaya sometimes, one tablet sometimes. So these folklore treasures or secrets, if we explore, if we establish, if it is useful, we can bring them to the regular use. If they are not useful, probably we can ignore them, right? Then coming to some advanced researchers, probably not for PG students, but as PG guides, we can think, or teachers for our career, we can think about these as a lifetime achievement of ours. As I discussed earlier, Arthava, analyzing it for its cytokines, all etc. Studies we can think about. Then changes in vaginal flora microbiome is the latest hot topic. Everywhere people are talking about microbiome, the gut microbiome, poo therapy, this, that. So with our Basti Chikitsa, what kind of microbial changes in the microbiome of gut flora is taking place? What is its impact on vaginal flora? Whether these two are reflecting each other, healthiness of each other are reflecting. So such studies can also be thought of as out of box, right? Challenges. Then Uttara Basti, a much said area, of course, tubal block here, there, endometrial receptivity, everywhere we talked about it. What is the extent of absorption of the drugs when we give? Whether a grata has better absorption, whether a taila has better absorption, whether a near taila has better absorption, what combination of taila, what a drug when it is given, what enhances the absorption of the taila, what enhances the absorption of the grata, all these are simply a mystery. Then coming to Garbhadana, DNA studies and epigenetic studies. Now our Ahara Viharas, our Aushadas are proving their efficacy not only in simple diseases. They claim their efficacy even in changing the gene expression, right? So if gene expression can be changed, DNA modification can be done, why can't it be tried definitely in modifying the quality of the fetus, modifying the abnormality expected in the fetus, and modifying the problems expected in the fetus? So this level of studies, probably it is right time now for us to good look into, right? So if a baby is identified as Down's baby at 5th month by giving some Swarna Prayoga, by giving some Bradvata Chintamani with gold, like drugs, can we modify its composition, genetic composition? Really a question, right? So quality enhancement of the child with IQ level modifications, maybe autism is increasing, as in the previous sessions it was being discussed. So hyperactive syndrome, attention deficit hyperactivity syndrome, these problems when it is increasing, can we change it? Now you can hear that at the 30s, people are becoming victim of diabetes mellitus. So can we prevent it through Garbhadana Samskara? Can some genetic modulation be done, right? Gene mutation, is it possible towards mutation? Can be from 2 ways. One is bad mutation, one is good mutation. Can we do some good mutations for the sake of people? Epigenetics, infertility, these are areas. Then coming to studies on experimental models of tumor pathology, we can have definitely studies on this. Cell line studies with Agni Ksharakharma, we have carried out the Agni Ksharakharma study, cell line studies. Then Shodhana and gene modification, as I discussed earlier. Reducing the lethal gene mutations can also be thought about. And coming to the surgical area, if we can design, instead of depending on the modern Shastra Yantras, if we can, as described in Sushruta, we are designing some Shastra Yantra Shastras. We are bringing them into practice. For us, those who have seen Doan's retractor, Sarpafana means it will be Doan's retractor, right? But for the first year, Sarpafana means he remembers the Sarpafana. Like a Sarpafana, if we can design something through Ayurveda and we say it as a Sarpafana, first generation may remember both previous name and current name, but the next generation, when the generation wise we are changing the name, the forthcoming generation will remember it as a Sarpafana, and modern people will also say that this is something Ayurvedic. Sarpafana, they call it as, right? So we can prove that we have the treasure of surgery also. I think with this, I'm coming to an end of my presentation. If I had taken little bit more time, taking it because the privilege of having the last presenter of the session, I really beg your pardon and apologize for making you to sit for longer time. Girish, should I conclude? Girish?
Yes, madam.
Okay.
Madam,
Yes. Yes, Girish.
could you please on the camera, madam?
Let me try. Yes.
Thank you, madam. Despite technical difficulties, I personally felt that it was a wonderful presentation. The presentation speaks about your depth and breadth of the experience in the field of Prasutitantra and Striroga, madam. It was a wonderful presentation. Hope all the delegates would agree with me. So there are several questions. I would, if you agree, I would like to two questions selected. What are the effect of external factors on growing fetus within the womb, especially with relation to mother?
Epigenetic modulation, that is the area of interest nowadays. In what way it is affecting the growth of the fetus, in what way it is affecting the health of the fetus, in what way it is deciding the future growth of the fetus. All these are explained through epigenetic. So
Yes,
external factors, earlier we used to say it is ahara vihara. Now how they are having efficacy is proved through the epigenetics. So it's a wonderful area which we can, I think we have to embrace this concept to explain our role of ahara vihara. We should not
Thank you,
leave
madam.
it as something boring.
Thank you, madam. One more question. To what extent Pumsavana karma is effective? Kindly elaborate.
See, Pumsavana karma, like earlier other subject I said, it is a double-edged sword. Whenever you say Pumsavana, it is known for its bad side, like getting a male child, because it has been practiced by many so-called practitioners to getting a male child. Instead of thinking of male child, if you concentrate on the other two areas which were said, the three indications out of three indications of Pumsavana, one is to establish, or those who are not attaining pregnancy, for them Garbhasthapanartham. Then those who have attained pregnancy but it is not becoming established or it is not getting sthira, for them Garbhasthirikaranartham. And the third intention is to have a desired child. So first two intentions, if you are using, that's very well. And maybe now, as I said, it is the nasal root working on HPO axis, influencing the fetal HPO axis, maybe changing its genetic modulation, whatever it is. So these areas are the unknown areas which you, as a researcher, we have to study. Not as a clinician, not as an academician, we can do the studies on this. Only as a researcher, if you dedicate your lifetime, end of your lifetime, you will be coming with, after diving into the ocean, you will be coming with one Ratna, but that Ratna is valuable.
True, madam. Thank you, madam. Last question is, what kind of research helps in Stri Shukra versus hormonal axis? That was the first part of it. Diet of mother, as it was already answered. So what kind of research helps in Stri Shukra versus hormonal axis? Maybe the methodological approach the delegate may be asking.
See, every action, hormonal balance or imbalance is working through H2 axis in females, right? Maybe Shodhana has a very fast action on regulating the hormonal imbalances or correcting the hormonal imbalances, as it is evidenced by many studies conducted on polycystic ovarian disease. We have concluded a study on 100 patients wherein we could see that the abnormal DHEA and DHEA, right? So these hormonal levels, those who are having abnormal, in those patients only, the abnormality has come down. Those who had normal LH, FSH, DHA, all these levels, they remained as normal. So hormonal abnormality can be very well tackled with the Shodhana therapy, followed by Shamanaushadis. So maybe that may be Shamanaushadis,
But since my practice is institutional, often I go for Shodhana therapy. Shodhana followed by Shamanaushadhis, and my experience says it is best.
Thank you so much for enlightening us with, you have, it was a very crisp and elaborate and to the point. I think it was, I believe that it's a wonderful speech, wonderful deliberation. Now I request Dr. Gayatri Bhatt, HOD and Professor, Department of Prasutitantra&Striroga at SDM College of Ayurveda Hospital, Hassan, to give remark on the occasion. Please, madam. Please unmute.
Thank you, Girish. Good evening. I'm very happy that I could hear my ma'am's talk completely, and very informative and detailed, the ideas about the researchers areas in Prasutitantra&Striroga has dealt by her, and especially she has highlighted mainly the issues like antenatal care, the importance of antenatal care, Prakriti, and Jataharani and Garbha Samskara really are very interesting research areas for researchers. As WHO already has taken one cohort study on relationship of maternal nutrition and on the preventing the the non-communicable diseases. It is already done in India, Canada, multi-country actually, South Africa, Canada, India, and China. And it's a cohort searching 1930 they are doing. So just like that, we also can do research taking this Masanusmasika Pathya and its relationship on the birth weight as well as in preventing the non-communicable disease. Because nowadays the whole public, the generation, young generation are getting hypertension, diabetes, such non-communicable diseases. And another thing she highlighted was Prakriti, taking a relationship of Prakriti. Also the research can be done like whether which Prakriti ladies attain menopause early or late, and also whether this Prakriti has the relationship on the labor outcome, whether which Prakriti, any difficult labor, whether Vata Prakriti ladies having a difficult labor, such observational study also can be done. And she also highlighted about the stanya, the stanya pariksha and all. Here also Prakriti, whether Kapha Prakritis, they are having stanya sampat, and all stanya dushtis are, whether it is more seen in Vata Prakriti ladies, such very small things but having more impact on clinical practice. Such researches also can be taken. And mostly very common problems nowadays in the antenatal visits we see are gestational diabetes. Once again, whether any Prakriti relationship, the Kapha Prakriti lady taking Abhishandhi Ahara, Santarpana, Brahmana during the antenatal visit, getting a GDM, this type of observational study also can be taken. Because during pregnancy, intervention by intervening with the drugs, doing research is really very difficult we have to prove the non-teratogenicity. Hence, this type of observational study will give us more, the authenticity, and also whether this Prakriti has, the relationship in pregnancy complications or labor complications. Such areas also can be done. She has given hint, or she has put a light on us to take a vast area of research taking the Prakriti. And also she said about IUGR, and also whether we can prevent IUGR, manage IUGR, such thing, such researches also can be done. And another thing she said about Jataharani, and as of course nowadays this COVID, whether this COVID can be taken as Jataharani, just because Jataharani, it in that description only says that it's Garbham Hanti, Vapum Shahanti, Jayam Shahanti, Jayamanam Shahanti. So the Jataharani kills the pregnant lady, child, its and it causes abortion. Whether we also know it is a burning that many pregnant ladies are getting COVID infections, and also we are getting one different news from different directions, corners, that pregnant ladies are more getting complications and they are dying. And also whether if the pregnancy during pregnancy comes negative also after the treatment, whether this COVID is having impact on the born child, maybe till its till its adulthood also, with a big cohort study can be taken this corona also. And whether this corona the infection, COVID infection during pregnancy, whether garbhini jwara chikitsa can be adopted, that also can be taken for research. And also she said about diet and lifestyle for reproductive health also. She said that was also area of interest, because nowadays we know that increasing prevalence of polycystic ovarian disease, endometriosis, and nowadays we all know that declining fertility. So whether this ahara, whether, because Ayurveda has told ahara vidhividhayatana, viruddhahara concept, all these things are told, whether adopting this whether adopting improve the quality of reproductive health, that also is one of the research area. And it's another thing is she said about the folklore practices should be taken for research. One of that is squatting for labor process. So when I was in London, I have seen that water baths. So my students, they were pregnant, 1 or 2 people. They had asked me to come to their house. I have witnessed they made one artificial rubber tank filled with water, and they delivered there. So whether such thing can also be done, because squatting position in the folklore practice is also very much helpful in the descent of the fetus. Whether such thing can be adopted. And as he said, mantra chikitsa in labor process is told, whether can it be the modified like hypnotherapy or such, or the counseling during the labor process, whether it will ease the labor the process, and whether it helps in the delivering help in the easing the labor. Such the studies can be taken. Overall, the whole 1.5, I think almost 1.5 hours, the talk the madam has given very beautiful ideas on the researcher. So the young researcher who are participating in this webinar, and I think hopefully they will take these ideas and they will take up some good research project in future. I thank once again, I thank madam for giving her that time.
Thank you so much, Madam, Dr. Gayatri, Madam. Again, thank Dr. Mamatha K V, Madam, Principal, SDM College of Ayurveda and Hospital, Udupi, for accepting our invitation and enlightening us with widely about the research areas related to Prasutitantra and Striroga, Madam. On behalf of Principal, the faculty members, and all the delegates, I thank you again. And I thank Dr. Gayatri Bhatt, HOD, Department of Prasutitantra and Striroga, SDM, Hassan, for giving her remarks. With your kind permission, I'll wind up the session. Tomorrow, dear delegates, we have 3 sessions. We have a presentation from morning 10 o'clock till 1 o'clock. At 10, we have got Dr. Pawan Kumar Godretwar, Dean Research, NIA Jaipur, on need for research in postgraduate studies. At 11, we have got Dr. Anand Katti, GAMC, Professor GAMC, Bengaluru, on components of synopsis as per RGHS. At 12, we have got a lecture by Dr. Suhas Kumar Shetty on sample size calculation and standard tests in synopsis writing. We thank all the delegates for their active participation And we anticipate the similar kind of participation in the next coming 3 days. Please take care. Thank you. Good day.